it. Moises was my PhD student. Actually, he was actually my first PhD student. Graduated from UT Austin. So even though we moved here 2011, when Moises moved, he stayed enrolled at UT Austin. So he did all of the coursework there and then came here and did all of the lab work here with me, and then graduated in 2015. We worked a lot with population genetics and speciation and hybridization, which are themes that he still works on today. But then after the PhD, he went to KAUST and then to State University of New York in Buffalo to kind of shift his research focus a little bit from more adaptation as it relates to speciation and evolution to adaptations as it relates to climate change. So now his main line of research is looking at climate change, the effects of climate change, and how populations are adapting to climate change, which is something really, really interesting. And most recently, he became an assistant professor at Auburn University and is setting up a fantastic lab. I was there. When was that, Moises? I think it was about a year ago, right? Maybe less than a year ago.
Yeah, okay. December last year, before the world changed completely.
Before the world collapsed. I think it might have been the last trip I did. It was the last air travel I did since the whole thing went down in flames. But anyhow, yeah. I saw the beginnings of the lab. I'm sure it's much more complete now, but everything I saw was fantastic, and I could not be prouder to see where my first PhD student ended up. So without further ado, I'm going to pass the torch to Moises and let him talk about his current research at Auburn.
Thank you so much, Luis, for this awesome introduction, and thank you so much, Athena and Alejandra, for the invitation as well. Well, thank you, everyone at the Academy, for those old friends that are here. I mean, probably the last time that I was at the Academy was 2015, so I'm very excited to be back, at least virtually, to see everyone. So thanks, everyone, for tuning in. So as Luis mentioned, I was there from 2011, 2012 to 2015 when I finished my PhD. And while I was there, we had a great opportunity to actually have some outreach activities. This was one of the favorite things to do at the Academy here with the famous Hudson Pinedo, who was another student of Luis and then postdoc. It was always great to test the equipment of the CCG. As you can see here, at least we were wearing safety goggles for that. Also, there were amazing interactions with scientists from E.O. Wilson all the way to fellow PhD students that were using the CCG as a tool to develop their genomic projects. So here we have Gary Longo, who was a student at the time with Professor Giacomo Bernardi, and was collaborating with us in some of those projects associated with genomics. So the Cal Academy was really foundational to my success as a scientist, and I owe a lot to the Academy. So I'm very, very grateful of all the opportunities and all the chances that I have to develop research here. So thank you, everyone, for attending. So I hope I don't have to convince anyone in the crowd that the climate is actually changing. So the questions right now is not whether it's going to change or not, but actually how much it's going to change. So depending on the emission trajectories, we're either going to be talking about increases that can be from 6 to 4 degrees Celsius by the end of the century, or something more associated between 0.5 and 1.5 degrees Celsius. And as you can imagine, this will actually have a tremendous effects on marine organisms because many of them actually do not regulate their internal body temperature. So in the case of marine fishes, which is what Luis and many members here at the Academy, and John McCosker too, thanks for coming to the talk, are really obsessed with, is understanding how these fish are going to respond to that. So we now know, thanks to a lot of work that has been done on this, that this can actually alter the developmental rates of many marine species, in this case, the Thalassoma lithosciatum. We can see here that it can also affect the aerobic capacity of the fish. It can cause reduction on swimming speeds, and it can cause all sorts of detrimental effects associated with reproduction, from changes in sex ratios all the way to diminished reproductive output. And this is a very active area of research. But one of the things that we also have to take into account is that these are a lot of times these are observations at the individual level that are done immediately. So because of these changes, overall, what we expect, some of these responses, what we're expecting is that over time, we're going to see adaptation of those heat-tolerant genome types where, you know, the population might actually shift from some genotypes that are more generalist or adapted to cold water towards warm water. And this might actually lead to a reduction of overall genetic diversity. We can have range shifts where, you know, there can be a lot of interactions that can be detrimental for both the invasives and the local species. And one of the most active areas, and what I consider one of the most attractive areas of study right now, is the possibility of acclimation via phenotypic plasticity. So when we think about this in the long term, what is going to happen from one generation to the next while these conditions are changing because of the emissions from fossil fuels? So one of the things associated with acclimation and phenotypic plasticity is that scientists have actually seen, and this is not only seen in fish, there's similar studies for oysters, similar studies in mice, Drosophila, plants, all sorts of organisms. And the idea is that if you subject a population to a stressor, in this case, we're talking about warming, those individuals that are actually exposed to that stressor are going to pass some sort of information, or it could be nutritional provisioning, hormones, or epigenetic marks to their offspring. So they actually do a lot better under those adverse conditions. Of course, there's also an element of genetic inheritance where, you know, if the parents were already naturally really good at surviving warm temperature, they'll pass those genes. But there's also going to be a component of non-genetic inheritance where just because these parents were exposed to adverse conditions, the juveniles might be way better off in those same adverse conditions. And this is something that has been pretty well studied in some marine fishes, some better than others. And some of the things that are very interesting to study is what are some of the limitations to these systems, because this doesn't always happen. And also, a lot of times what we assume is that the conditions are going to be the same from one generation to the next. But there's a lot of questions associated with what happens when the environment changes between the parents, their offspring, and what are some of the dynamics that those changes in condition can take, especially over the juveniles. And that's a lot of what I'm going to talk about today is associated with that. So the three research questions that I'm going to be discussing today are presented here, and I'm going to start asking the question, what happens when you change the conditions between generations? Is this going to disrupt the transgenerational acclimation? And this is a paper that came out a couple of years ago now in Molecular Ecology, where an experiment that had been done by Jennifer Donaldson and Phil Mundy back in 2009 or 2010, they had already had the setting for this, and I came more for the analysis of the genomic data and a little bit of the physiological data. The idea is that fishes were set out in the wild. Fishes were collected from the wild. They were brought to an aquarium. They reproduced in the aquarium, and then once they did that, they were set up into different temperature conditions. So the fishes could either be at control, the F1 was either at control, plus 1.5 or plus 3 degrees Celsius. And they were maintained at those temperatures for 2 years. Then after that, these fishes reproduced in the lab, and then the offspring, the F2, were subdivided into different conditions. Either control, developmental, plus 3 degrees Celsius, transgenerational, plus 1.5. And then you can see here that there's a step treatment, and this is the star of the show, because the grandparents were at control temperature in control temperature with respect to the Great Barrier Reef. The parents were at plus 1.5 above the average of the Great Barrier Reef, and they themselves were at plus 3 degrees Celsius. So this represents a stepwise increase in temperature. The idea here is that we were comparing this to juveniles that actually come from a lineage at plus 3 degrees Celsius for 2 generations. So the question is, what effects are we going to see to this stepwise treatment compared to control treatments, compared to transgenerational at medium temperatures and transgenerational at really high temperatures? For this, we measured phenotypic measurements like liver weight, body weight, and then oxygen consumption at both the resting and the maximum activity. The resting is that you literally put the fish on an acrylic chamber, you measure the resting oxygen consumption. For the maximum activity, you have an acrylic chamber, but you put a steering bar inside. The fish gets really stressed, and then you get it when you get the respiration rate when it's really, really stressed. And then the molecular responses, which were pretty much associated with the differential gene expression of the liver. And I'm not going to go into details about this. If you're interested about it, we can discuss it, but these are pretty standard methods. And they didn't have anything, completely novel in the way that we were developing them. So one of the things that we found that was very, very interesting is that for those fish that actually were set up to elevated temperature, in this case, +3 degrees Celsius, they showed an enlargement of the liver with respect to the body size. In fish, liver is usually associated with an increase of lipid storage, but in this case, Acanthochromis polyacanthus did not show any fat in the liver. We actually think that this was associated with processes of detoxification and immune defense as a response to that really, really intense increase of temperature. So what was interesting is that, and I'm going to mention this at the end of this section, we also found genes associated with inflammation in all of these treatments. So this was a result that has not been reported in fish before, the liver size increase, but it has been reported for rats and mice when they're actually exposed to a lot of aerobic activity. So there is some association there. So it's not completely unexpected, but it hadn't been reported before. In terms of the aerobic capacity, so the aerobic scope is just a measure of the difference between the maximum oxygen consumption and the resting oxygen consumption. And the idea there is that that difference that difference between the resting and the oxygen consumption rates is the amount of energy that an individual has for their daily activities. And you can see here that the aerobic scope of the control is equivalent of that of all the transgenerational treatments, even the one that had the stepwise temperature increase. But you can see here that the fish that are seeing the warm temperature for the first time, the developmental +3 and the acutes, which were only exposed to those temperatures for 10 days, did not show compensation. So we can see here that even if the temperature increase, when the temperature increase happens from one generation to the next, these individuals might actually still be able to compensate for that. So we did the analysis of gene expression using as reference the genome of Acanthochromis polyacanthus. So this was roughly 25,000 genes that we were able to analyze. And the idea here is that we're comparing the stepwise treatment to everything else. And one of the things that was interesting is that between step and control, we actually found activation of genes in step that were associated with energy levels, regulation of cell cycle progression, immune response, and mitochondrial activity. And mitochondrial activity is something that I'm going to keep repeating over and over during the talk, because a lot of times when you see a lot of these heat stress, it's actually associated with the increase of oxygen consumption, and the mitochondria gets really, really activated as well. What we saw is that the genes that were regulated in step associated, that were compared to the other two transgenerational treatments, we saw that step had much more mitochondrial activation, detoxification, and genes associated with stress response. And something that was very interesting is that for the transgenerational +3 degrees Celsius and for the step +3 degrees Celsius, we also found that there are genes associated with inflammation. And these are as I mentioned earlier, these were the treatments that had the larger livers compared to the rest of the body size. So we have both the phenotypic and the molecular evidence for this liver expansion. So in conclusion, it's interesting that the transgenerational treatments, all of them were able to compensate their aerobic metabolism, but the step treatment doesn't do it in the same way. So it is possible that metabolic compensation will take place when fishes actually see warmer waters than their parents, but the acclimation process is very different from both a phenotypic and molecular perspective. And as I mentioned, there's more details in the paper that was published in 2018. Now, for the second objective, we really want to understand, too, what happens when the temperature changes at the time of reproduction. Is that going to cause a disruption in these transgenerational acclimation mechanisms? So you can imagine a scenario where, you have a parent that develops at 32 degrees Celsius. When it comes time to reproduce, the water unexpectedly changes to 25 degrees Celsius, but then the offspring are again subject to 32 degrees Celsius. So what is going to be more important? The influence of the temperature at which the parent developed? Or is it the temperature at which the parent reproduced? Or is it a mismatch between the reproduction and the juvenile development that's going to cause stress? And again, I have to give credit here to my collaborator, Jennifer Donaldson and Philip Mundy, because they were the ones who did this. And this is probably 10 years of experiment, because each one of these different color brackets represents 2 or 2.5 years. So I think this experiment started in 2008, where you can see here that fish were brought from the wild. They were, again, reproduced in captivity. These F1 generation was divided into 2 treatments, either control or plus 1.5 degrees Celsius, which is more or less a medium scenario of warming for the year 2100. These black boxes represent, again, the division of another generation. So for the F2, you can see that the fishes were developmentally maintained at that control temperature. And here they were maintained at plus 1.5. And then 6 months before the time of reproduction, the fish were either maintained at the same temperature, and another section of the breeding pairs were actually set up to a different temperature, in this case, plus 1.5. And you can see here we did the same. So this is a mirrored experimental design, where this lineage is here at plus 1.5. At the time of reproduction, they were either maintained at plus 1.5 or they were divided to control. And then the same for the juveniles. The juveniles were either split at control or to plus 1.5. And this very ambitious experimental design allows us to understand what happens when you have a developmental exposure to warm temperatures, so when you're seeing it only during your lifetime. What happens when the reproduction of the parents and you yourself match that warm temperature? And what happens when it's a full transgenerational effect? So what happens when you have these full complete exposure to warm water from your grandparents, your parents' development, the parents' reproduction, and your self-developed at plus 1.5. And we again measured oxygen consumption and gene expression. So one of the things that we actually found is that the maximum oxygen consumption, so the maximum capacity for a fish to actually consume oxygen, is actually really, really influenced by the conditions of the grandparents and the parents. So if your grandparents and parents were actually exposed to warm temperature, in this case plus 1.5, you actually have a better capacity to tolerate that warm water. And this represents a true transgenerational effect, because that was an observation that held on irregardless of the conditions that you had as a juvenile. And this is a very important finding, because a lot of times critics of transgenerational plasticity suggest that everything is controlled by development. And we're seeing here that there is some effect from the grandparents and the parents as well. Another thing that we found that it's actually very interesting is that there's actually, if there is a mismatch between the temperature of reproduction of the parents and the conditions at which the third generation juveniles are experiencing, you're actually going to see an increase in the resting oxygen consumption. And I'm going to show this also from a molecular perspective, because from the molecular perspective, we also captured it. So we're already seeing that here, the temperature of the grandparents and parents has an effect, but there's also an effect of the developmental temperature and the reproduction of the parents. When we look at this from perspective of the differential expression, again, using the genome of Acanthochromys polycanthus, we see that in this case, these are the fish at control. So that's a blue fish represents that the juvenile was in control conditions. The inner circle represents the conditions of reproduction of the parents. In this case, the parents reproduce at control. And the outer circle represents the conditions of the grandparents, okay? So we can see here that if you were always at control transgenerationally, or if your parents and you yourself developed that control, there was not much activation of genes associated with stress. On the other end, if your grandparents and your parents were at control, at warm temperature, your parents reproduced at warm temperature, and you yourself grew up in an environment that was control water, so it was much colder water, there's a very intense signature of its stress. And you can see it right here because the amount of differentially expressed genes between all these between these categories is an order of magnitude higher than between these two categories. So what we're seeing here is that what is actually really determining the resting oxygen consumption and the measurements of gene expression is actually the match between the conditions of the juvenile and the reproductive temperature of the parents. And what's really beautiful about this experimental design is we can also see it from the perspective of the warm temperature, and the results are pretty much the same. We don't see a lot of activation of stressful genes for those fish that had a match between the reproductive temperature of the parents, the juvenile temperatures, and we do see a lot of stress for those fish that are seeing warm temperatures for the first time. When we look at the molecular mechanisms that are being activated, so if you're seeing these temperatures for the first time, like this fish here, you actually are going to have a lot of activation of genes that are associated with cellular stress, like oxidation reduction processes, respiratory chain complex, catabolic processes, and lipid metabolism. So you're pretty much consuming everything you have, and you're trying to deal with that excess of oxygen in all your tissues. And then if your parents reproduced at plus 1.5 and you yourself were at plus 1.5, or if you were completely transgenerational plus 1.5, the molecular mechanisms that stand out actually have to be with growth, protein synthesis, and cell division. So that means that these individuals right here are thriving. They're not dealing as much with all these processes associated with stress. So in conclusion, the exposure of previous generations can actually lead to an increase of oxygen consumption, and that is in a sense a true transgenerational effect. In a sense, what we're seeing here is that the parents are actually preparing juveniles for the conditions that are present at the time of reproduction. And then it's important to recognize that to activate the metabolic compensation, the juveniles actually have to develop in an environment that is relatively similar to the conditions of the parents. And this is a paper that is submitted, so finger crossed this time goes around. We try to submit it to some really large journals and no luck so far, so hopefully it goes this time. So the last question is more associated with what are some of the effects that marine heat waves are going to have on coral reef fishes? So one of the things that we're seeing these days is that marine heat waves are actually increasing in both frequency and intensity. And this is a picture right here from a bleaching event in the Great Barrier Reef in 2016. When I went to undergrad, that was not a crazy long time ago, my professor of marine biology, Luis de Cruz, always told us that the biggest El Nino event was that from 1997. And we can see now that there's been heat waves that are much, much warmer. And not only that, but that the heat waves that are coming up are much more frequent than they used to be in the past. So since the 1980s, it's estimated that the number of heat waves have actually doubled, and they're also increasing a lot in intensity. So in this case, we actually were wondering, this bleaching took place in the Great Barrier Reef. And with collaborators at James Cook University in Australia, we were actually able to pick up a lot of multiple species of fish at different times of these heat waves to try to understand how other marine organisms are responding, other organisms that are not coral. So the species that we selected for this project, just because they were relatively easy to collect, were Oysterhynchus doderlini, Oysterhynchus cyanosoma, Chilodopterus quinquilineatus for the apogonids, the palmocentrids, which were Palmocentris molossensis, and Acanthochromys polacanthus. And as you can remember, these Acanthochromys polacanthus is the same fish that I talked about in all the other experiments. So in a sense, we know how it's going to behave in captivity, but also now we also have a reference of how it's going to respond in the wild to a natural heat wave in that sense. So these are the temperature profiles, and I call this the confetti graph. And the idea here is that you can see that this pink purple line represents the heat wave of 2015, 2016. You can see here that December was already really anomalous because the temperatures were a little bit colder than other years. February and March was when the temperatures were the highest. The average was about 31 degrees Celsius. And then you can see that July, the temperatures start getting a little bit colder. Remember, it's the southern hemisphere. In July, the temperatures get a little bit colder, but you can see that there's a significant difference with the other years. So it is much warmer than other years. So thanks to our collaborators, we had samples from December, which is before the heat wave. For February, we had samples of 2 of the species, not all 5, because not all the samples worked out. And in March, we collected all the 5 species, which is the peak of the heating. And we also had samples for July, kind of like the recovery period, if you may call it that, when the temperatures were already getting lower. And you can see here that these temperatures are not that different. So we're talking about going from 27 to about 30, 31 degrees Celsius, which is not a crazy amount of variation. So for this, we collected 5 individuals per species per month. We didn't have samples of February for the apogonids, unfortunately. We extracted the liver of the fish. We sent them out for RNA extraction and sequencing. We did the de novo transcriptome assembly for most of the species. Before this project, 4 of those species didn't have any genomic resources. And this was a really big point of discussion. We actually wanted to compare the same genes, or more or less the same genes, across the 5 species. So what we did is that we actually selected 16,000 genes that were actually shared between all those lineages. And we mapped the RNA-seq reads back to those orthologs, and we did an analysis of gene expression and also analysis of correlation networks. I'm not going to discuss much the correlation networks today. I'm going to focus more on the gene expression. So one of the things that we noticed right off the bat is that when we do the pairwise comparisons between the different months, so here you can see December versus February. Again, the apogonids were not collected in February. That's where there's NAs in parts of these plot. December versus February and December versus March and so forth. We can see right off the bat with this plot that not all species responded the same way. So Acanthochromys polacanthus and Palmocentris molossensis actually were the most responsive, were some of the most responsive species. But on the other end of the spectrum, we have Chilodopterus quinquilineatus, which almost didn't respond at all. And you can see here that the response between the 2 months with the most different temperatures, the average temperature of March was about 31. The average temperature in July was about 24, 25, so it's 6 degrees Celsius of difference. You can see here that for the other species, there were hundreds, if not thousands, of differentially expressed genes, and for quinquilineatus, there was only like 20. So the other species actually had a more intermediate response. And something that was very interesting for those individuals that we found, that we actually managed to collect in February and March, which is Acanthochromys polacanthus and molossensis, is that we actually saw differences in the patterns of gene expression between February and March. February and March are actually very interesting because they represent, February represents the beginning of the heat wave, whereas March represents the intermediate portion of the heat wave. But they pretty much have the same temperature, around 30, 31 degrees Celsius. So we see when we do this principal component analysis, what we see is that the patterns of gene expression are different for the cold months, July and December, and the more warmer months. And the other difference here for Polyacanthus is the difference between February and March. And we think that has to do with the fact that different genes are getting activated at different times of the thermal stress. And this is something that is really important because a lot of times when you have a heat wave event, we were limited on collecting only one data point from that, and we can compare like before and during the heat wave. But in this case, we had 2 points during the heat wave, and we can see that there's molecular differences between those 2 points. You can see here the same for Mollisensis, where in February, the samples are kind of all over the place, but in March, they're very tightly clustered because of that duration of that heat stress. So the responses are different at the beginning of the heat wave in February versus the intermediate stages of the heat wave in March. We can also see that with the activation of the heat shock proteins, for example. So this panel here represents a heat map where orange colors represent activation and blue colors represent more the deactivation. December, remember, was just kind of neutral temperature, even a little bit cold. February represents the beginning of the heat wave when they first see the heat for the first time. And you can see here that the heat shock proteins in both species get very much activated. But then in March, the temperature is still really high, but they don't maintain the activation of those heat shock proteins. And that has to do with the fact that the heat shock proteins are relatively hard to maintain. So this is not a secret. It's not like we came up with this, but it just confirms that the patterns of expression that you'll see at the beginning of the heat wave are different than in the intermediate stages of the heat wave. More importantly, the genes and the molecular pathways that we saw activated with the samples in the wild actually overlap with the laboratory experiments that I was talking about at the beginning of the talk. So Acanthochromys polyacanthus is probably the reef fish that has the most studies on climate change from both temperature, CO2, physiology, behavior, and genomics. And it has fueled the career of many scientists, including my own. So we know really well what happens when the temperature increases for Acanthochromys polyacanthus. And we know that this leads to the activation of mitochondrial activity, detoxification, fatty acid metabolisms, and glycogenesis as well. And we saw the activation of these molecular mechanisms for most of the species, except for one, Chilodopterus quinquelineatus. We didn't see any activation of stressful response for that fish, which is very interesting. So in the end, we can say that the heat wave of 2015, 2016 actually had a very intense effect on molecular responses. We actually saw differences between the initial and the prolonged stages of warming, and the molecular processes were shared between some of the species, and these actually have some concordance with previous laboratory experiments. And this is a paper that got out earlier this year in Science Advances, and we've got some pretty good feedback based on that. And it was a very sizable collaboration between KAUST in Saudi Arabia and James Cook University. So before I finish, I just wanted to mention a little bit of the future directions now in my lab. So for the past year, I have been working as an assistant professor at Auburn University. And some of the questions that I want to analyze is, how is the response to warming going to be modulated across latitudinal gradients? As you can see here in this plot, we have a lot of species that actually can be distributed across the Gulf of Mexico that are also distributed all the way to Massachusetts and even Canada. And you can see here by the profiles of these graphs, these represent the temperature, average temperatures for 5 years. We can see here that, first of all, not all the environmental parameters are going to be the same in the different sites. There's going to be historical population subdivision between these sites, especially between the Gulf of Mexico and the populations in the southeast of the US, between Florida and North Carolina. There's a natural barrier here because of Florida. And on top of that, the temperatures that are experienced by populations all the way far in the north are way different than those experienced in the Gulf of Mexico. So in this case, one of the things that we're really excited to work on is work on these fish that have these really broad distributions and have potentially intermediate potential for dispersal. And in this case, we're really excited to start working with an emerging model or a species that we want to develop as a model, which is the pinfish, Lagodon rhomboides. For this, we actually have preliminary data on how they behave in terms of temperature. So we did an experiment this summer where we collected juvenile individuals of pinfish and we exposed them to warm temperature for 10 days. So you see here the fish, these acrylic chambers are the acrylic chambers for oxygen consumption. You can see there's a red arrow here that represents the position of the fish. And then there's an oxygen sensor that allows measuring the amount of oxygen in the water that leaves the chamber. And these measurements are taken over periods of 24 to 30 hours. So we have a really good, reliable measures of the oxygen consumption at rest. We don't have the equipment yet to do it, the maximum resting oxygen consumption, but we hope to do it in the future. And what we can see here is that when we subject the fish to the average temperatures of 26 degrees Celsius, the average temperatures of the estuaries of the Gulf of Mexico, we find that the fish has this oxygen consumption, and there's a significant increase in the resting oxygen consumption at 29 degrees Celsius. And what's very interesting about this measurement is that if you increase the temperature even more, the resting oxygen consumption doesn't increase even more. Okay, so we're seeing here that after 29 degrees Celsius, that species reaches a limit in which if the temperature keeps increasing, it will not be able to sustain its aerobic metabolism. So we were really surprised by this because everyone told us that, oh, the pinfish is just going to survive everything. If you throw it in boiling water, they're going to make soup with it and then eat the soup and then just go back to the water. Nothing's going to happen. But we actually are noticing that the pinfish is much more sensitive to warm water than we anticipated. So this is a model that we're going to try to start working with because it's one of the species that's the most important links for food webs in the Gulf of Mexico. Everything eats pinfish, from dolphins to pelicans to groupers, everything eats the pinfish. So it's a very, very important species. And hopefully we can get funds to keep working on it. One of the things that, and I put this slide here specifically for Brian Simonson, one of the things that we have been doing is that we already have developed an annotated transcriptome, and my grad student is working on that publication. And the genome is approximately 1 GB, which is not very big. We have that advantage. And in collaboration with the Crab and Hub Lab at Sunny Buffalo, we've actually been developing a genome with long-read sequencing using the Oxford nanopore approach, using an equipment called the GridION. And for now, we've been obtaining these really amazing results where the Oxford nanopore actually spits out sequences that are already 140,000 base pairs. So just for references, the Illumina machines usually spit out fragments of 150 base pairs. When you do Sankara sequencing, those fragments the maximum can be like 1,000 or 1,200 base pairs. So now with this Oxford nanopore approach, you can get on average reads that are 50,000 base pairs. We've already done 2 preliminary runs to assess the completeness of the genome, and we're actually in pretty good shape. So we actually now have a genome that has a 28x coverage for the pinfish and an N50 of 1.8 megabases. So now the next steps are going to be to complement this with Hi-C to actually get a chromosomal scale genome for the pinfish. And that's actually going to be one of the chapters of the dissertation of one of my PhD students. So we already have the transcriptome that's also going to help us with the annotation. The other thing that we're doing is that we finished this July, in the middle of the pandemic, we actually finished the development of our own aquarium facility. So this is in the basement of the biology building, and this aquarium room was full of trash. It was a deposit, and we managed to refurbish it. And we actually have these 6 independent towers that have 2 shelves of aquaria that are actually going to allow us to do experiments of temperature, both for cold stress or for warm water stress, and also oxygen stress. And the idea is to collect individuals in the wild in seagrass environments of the Gulf, bring them in the lab, and then ask questions associated with thermal stress to understand what is the potential of developmental and transgenerational plasticity in some of these species of the Gulf that are of great ecological and fisheries importance. So as a take-home message from my talk, I just wanted to say that acclimation of marine fishes is possible, but it's very beneficial when the conditions of the parents are relatively similar to those of the offspring. It's very important to consider heat waves, because heat waves that are becoming increasingly frequent, are increasingly intense, are going to cause really intense disruptions to some of the patterns of gene expression and the metabolic capacity of these fish. So we have to consider heat waves, because it's not only the gradual increase of temperature, it's also going to be the thermal fluctuations. And then also we have to take into account the differences across latitudes. For some species might be more important than others. And there's been already a great amount of studies done for this in corals. There's been some studies in fish, but the studies in fish a lot of times kind of assume that, you know, dispersal is infinite and that there's no barriers. So I think we're in a really good position to understand these dynamics of adaptation and acclimation, where there might be historical population breaks, for example, in the Gulf of Mexico. And with this, I wanted to acknowledge my collaborators, Tim Ravasi, who's now at the Okinawa Institute of Science and Technology, was my supervisor in during the PhD, and during my postdoc, and he was one of the forces behind all this transgenerational studies. Jennifer Donaldson, who was a collaborator for this and the brains behind this. Phil Mundy as well. Celia Schunter, who was involved with the Heat Wave project. Trevor Krabenhoft, who was my supervisor for the second postdoc that I did, and is also integral for the development of these genomes that we're working on. Auburn, of course, because it's paying for part of this research. Also, this research was financed by JCU and KAUST in Saudi Arabia. And then also a final message that we're actively looking for grad students. These are my current 2 grad students, Katie and Logan, who are developing a lot of these questions. And this is my technician, Adam. So we really want to add more members to this team to try to understand, you know, local adaptation, acclimation, plasticity, and so forth. So if you know of any master's or PhD students that are interested in this sort of research, please contact me. And then I'll just leave room for questions now. Thanks for your attention. Yeah, and Alejandra, Luis, if you can dictate the questions of the chat, because I can't see them, I'll be happy to answer them.
All right. Thank you so much, Moises, for such an amazing talk. It's really a pleasure to have you. Brian saying clap, clap, clap. It's very exciting to hear that fish actually had some capacity to tolerate, you know, mild changes in temperature. All right, everyone. Any questions for Moises? Annika Kapan. Oh.
Yeah,
it's Durrell.
Go for it, Annika.
For some reason, the last Google user gets the ownership of that. Okay, so from Annika to you, Moises, great talk. And a quick question about microevolution. So in the experimental design for your 2018 paper, it ensures non-differential fitness effects, right? You're not taking only the surviving offspring, are you?
Well, first of all, there was not a lot of mortality. So
Right.
that's something that we get asked a lot, is the mortality, because mortality is a very big influence for a lot of these studies, but let me go back to it. But in this case, we didn't see a lot of mortality between all these different groups. One thing that was different, though, is that the control in the plus 1.5 actually produced a little bit more offspring than the plus 3. So there is an effect, there is a breeding effect there, where the pairs don't produce a lot of offspring. So it's not mortality, it more has to do with the number of offspring. So there could be an effect of selection. Yes, it's not just plasticity, there's also an effect of selection, because not all the breeding pairs do really well at plus 3 degrees Celsius. That's correct.
Okay, so turning that around, what is the potential for microevolution to capture some of the plastic responses and build upon them, given the different conditions you simulated. Some of these conditions are like this, and some are like, woo.
Yeah. So I think, I mean, that's another very interesting area of research, because now we've gone from like saying that phenotypic plasticity doesn't exist to saying that plasticity can actually be selected for, and it can actually, there's actually genotypes that might be more plastic than others. Number one. Number two, there can also be, these are not mutually exclusive. So you can have both plasticity and, you know, evolution taking place, selection taking place from time to time. So it's not like one or the other is going to take place. They're both going to take place. But in a sense, when you think about it, there are a lot of limitations for plasticity, which I've mentioned here, but there are also limitations for the effectiveness of selection, because when you select only the ones that do really well at really warm temperatures, a lot of studies have demonstrated that a lot of theoretical simulations have determined that when you do that directional selection, you actually reduce your standing genetic variation. And by reducing the standing genetic variation, you're more vulnerable to future stressors, right? So you know, plasticity might be limited in the sense that not all the phenotypes are plastic, but then selection might be limited because you're leading to a reduction. Ultimately, you might be leading to a reduction of diversity in that population or in that species. Yeah.
So yeah, that's something to think about. Great. Thank you.
Thanks for your question, Andrew.
Moises, you are receiving more claps from Lindsay Palaima.
Oh, hi, Lindsay. Thanks.
Any other question for Moises? Well, while someone else thinks about another, I do have a question, and it's related more with the species that you are using. So I was wondering, as far as I know, the Acanthochromys, Polyacanthus, and in general Acanthochromys, I think they are territorial, right?
Yeah.
So it's my question, if you think that repertoire of like the molecular repertoire of fish that they are territorial, maybe broader than for those fish that can move further away and maybe reach deeper depth and in a cooler space. Or if you think that, I know like maybe those fishes that are like sometimes bigger, it could be harder to breed in captivity. Mm-hmm. But if you have any hypothesis about how that repertoire can change across taxa.
Yeah, so I know that I can't see the screen right now and I can see the camera, but I know Luis is laughing right now because that's the question he always likes to ask. Because this species, Acanthochromys polyacanthus, has direct development. So actually the juveniles just, you know, come out immediately as kind of a tiny fish, and they don't have a larval duration. So it is possible that plasticity is much more preferred in this species because they don't have the, you know, they don't have to go away to a very different location that the parents are not, you know, where the parents are not. So when you actually look at the population subdivisions across the Great Barrier Reef, there's actually significant differentiation between the different populations of Acanthochromys polyacanthus. So every time we present this at a conference where there's fish people, everyone's always like, haha, but the reality is that very similar mechanisms have been found in other fishes. So not only in Acanthochromys polyacanthus, but also now there was a paper by Phil Munday that came out on kingfishes, which kingfish, honestly, like they're one of the fish that have the capacity to swim maybe all over the world. I don't know if John and Luis want to say something about that, but kingfishes just go everywhere because they're pelagic fish. And similar mechanisms are found in the kingfishes. Similar mechanisms have been found in experiments with the clownfishes as well. And part of the predictions of what we're seeing is that in terms of the theoretical methods that have been put out there, when you have species that don't move very far away from the parents and they don't have a lot of larval dispersal, what's interesting is that it's possible that transgenerational plasticity might be preferred. So in this case, it's possible that we're seeing transgenerational plasticity because the parents are there, the juveniles are going to be right there, and they're going to have the same conditions. So the transgenerational effect is really big. In other species that have broader pelagic, larger pelagic larval stages, for example, the pinfish, the larvae spends a lot of time in the water and spends a lot of time in the Gulf of Mexico until it reaches the estuaries, and that's one of the reasons why I chose it. What's interesting is that in those cases, it's possible that developmental plasticity is what's important, because the parent is over here, the larvae is going to go up in the water, and the conditions might change throughout the lifetime of that organism. So these are some of the predictions that, you know, we can play, and some of the predictions that we can actually test with some of the limitations that these fishes have. Because one of the advantages of Acanthopholis polyacanthus is that it's super easy to breed in the lab, and it's really easy to just grab the juvenile fish from one point to the other. But now we have to start asking these questions and fish, maybe not from a transgenerational perspective, but starting with a developmental perspective. And that's exactly why I'm interested in working in the pinfish here in the Gulf, because the juveniles, the parents are in the ocean, and the juveniles then go to an estuary. So I really suspect that there's going to be a really intense developmental scenario going on there, which is going to be different to the polyacanthus. So it's not one size fit all in terms of acclimation. You can have both transgenerational and developmental, and that might be associated to the ecology of the species.
Fantastic, thank you. I may see, I see Dr. John McCosker. I don't know, Dr. McCosker, do you have a question? Yes, please, go ahead.
Hi, Chair Moises, fantastic. And
thanks, John.
You're doing wonderful things. I'm wondering what the longevity of a reef fish, like these damselfishes, these pomacentrids that you're studying, the apagonids, as compared to a reef grouper or a moray eel, a long-lived fishes.
Yeah, I mean, that I'm not entirely sure, and then now I'm scared of screwing it up because it's you asking the question. So I don't want Dave Catania or Luis to grill me later. But I mean, yes, the size of these fish and the turnover of these fishes is also going to play an effect. So in this case, these pomacentrids, they probably live to 4 or 5 years, 6 years maybe in captivity. And then for the case of the groupers, you know, you can have some groupers that live 30, 40 years. And that's something that is also going to play an effect in how they respond to warming, because these species that have a higher turnover, like for example, blennies, there are some species of blennies and gobies. I was talking to Ross Robertson about this a couple months ago. There are some species of gobies that he bred in the lab, and the generation time was less than a year in Panama. I forget the group, but he bred them in the lab and in Naos Island, and he said it was less than a year. So when you have this really high turnover, it's possible that the mechanisms of natural selection might operate more successfully because you have such a high turnover that selection might operate better. But when you have these really long generation times, it's possible that plasticity is going to be more important too, right? Because the generation times are so much longer, and the number of individuals produced is also smaller. So it's possible plasticity is going to be more important in that sense. Yeah, great question.
Well, now that I've got the microphone, well done.
Thank you. Thanks, John.
Moises, I have a question from Matthew Badam. He says, do you think chromatin remodeling could play some effect in the heritability of these traits, or some other mechanisms?
Yeah. So one of the advantages and the disadvantages of working with this is that there's multiple people involved, obviously. So this sort of experiments, we actually saw it from many directions. So there's also a behavioral component. There's also a reproductive component to this. And then before I got to work in Saudi Arabia with these sort of projects, there was actually a group that was already working on the methylation. So the patterns of methylation actually do show that there is differential methylation between the different treatments, between the different transgenerational temperatures. So there are differences if you are transgenerationally exposed to elevated temperatures or if you're developmentally exposed to warm temperatures. And the idea is that that differential methylation could be associated to the differences in expression. Now, when we analyze the overlap of methylation versus gene expression, we only see about a 30% overlap between the two of them. So genes, 30% of genes that are both differentially methylated and genes that are differentially expressed. So methylation could play a role in it, but it's not the definite. It's not the definite response, not the definite answer. So one of the things that is happening with this project, now there's other postdocs that are taking on in this, and some of the things that we're trying to see is whether retrotransposomes are actually playing a role, because apparently retrotransposomes are playing a really big effect on how humans respond to stress. So it's possible that the organization of these genetic variants may actually play an effect. And the preliminary data that I have been able to see is that there are differences in response based on that. But that's some really deep bioinformatic work that I'm not involved with at the time, but hopefully it'll be published soon. But it's from the same group and from the same sort of experiments. Yeah, so that's the research that Professor Rabasi is doing now in Okinawa. Yeah, good question. Thanks.
Luis?
Yeah, I don't know if it's going to be quick or long, but Alejandro definitely stole my question. I always ask that about Acanthochromis because it's a direct developer. Anyhow, but that was not my question. My question is, now that you're kind of on top of this literature and you're actually working on it, how optimistic are you about the future of fishes? Is it about the future of corals? I am not optimistic at all. I think corals are going to be hard hit. It doesn't matter what we do, even if we control global warming. I think it's too late for a lot of corals up there. But I tend to be a little bit more optimistic about fish. But I haven't been on top of the literature, and I don't know when we see the really big changes happening. Right now here in San Francisco today, it's 90 degrees.
Yeah.
So when we start seeing that, more and more heat waves in the ocean, how do you think fish are going to respond in general as a whole group, like corals are?
Yeah, so I think, I mean, the thing about it is that these mechanisms actually have limitations. So when Jarrell asked the question, one of the things is that selection takes time, and also you can have reduction of diversity. But the reality too is that plasticity also has limitations. Just to give you an example, when we look at these traits in terms of reproduction, when we try to breed. So the breeding of the fishes that are set at +3 degrees Celsius, they breed in one generation. But then the second generation, we have an experiment where these fish were not able to breed the +3 degrees Celsius. So we can see here that even if they recovered their aerobic metabolism, and if they were able to recover their aerobic metabolism, they're not able to reproduce. So there are limitations to the effects of plasticity. I think a lot of the responses are going to be species dependent. So as you could see with the effect of the heat waves, not all populations are going to respond the same way, but also not all species are going to respond the same way. And this is something that the reviewers in this paper were grilling us about, because we really didn't have a good predictor of why Chilodopteris quinquilineatus was not having any effect. The only idea that we could have is that it was the species that had the broadest range. So Chilodopteris quinquilineatus, believe it or not, or like maybe if we go to the Red Sea, it's going to be a different species, but ideally supposedly is distributed from the Red Sea, Mozambique, all the way to Japan, I think. Or going all the way through the Indo-Pacific Ocean. Whereas Acanthochromis polacanthus is in a much more reduced area, right there between the Great Barrier Reef and some other adjacent areas of the Coral Triangle. So one of the things that we had is that, well, maybe there's a correlation there where, dispersal could be playing an effect, your capacity as a fish could be playing an effect. So I think a lot of these species are going to make it, but the most sensitive ones might be very endangered, especially if you have a very reduced range, if you have low capacity for dispersal, and if you are really close to your physiological limit. So those are the traits to your aerobic limit, because a lot of these tropical species, much more than temperate ones, a lot of these tropical species are already really close to their thermal ceiling. So an increase of 2 or 3 degrees could be really disastrous. Like the fish is not just going to die, but it might not reproduce, or it might only produce 2 offspring instead of 100. So I think, just to answer your question, like there's ways of predicting this, and there's going to be selection, adaptation. There's natural capacity for some fish to do better than others, like the case of Kinkelleniatus. And the highest risk factors are going to be for those species that are bad dispersers and that have really small ranges. So if you have a species that has a very small range in Belize, for example, like that endemic that you guys were describing, that would be extremely, extremely worrisome. And those are the species that would be more affected. The ones with really broad ranges, I would suspect that would be less affected. Yeah.
Thank you, Moises. Okay, so if you go, stop your sharing your screen, you will see I have lots of messages, and I can tell you're
awesome.
deeply missed at the Academy. Before everyone leave, I just want to invite you for next week, same time, same place. We will have another amazing speaker, Dr. Taryn Foster from the Albright's Lab, and she will be talking about the project she developed here at the Academy over the last year, I think. Thank you very much, everyone, for staying until the end. Moises, super happy to have you. Hope we can see you in 3D soon.
Yeah, thank you for the invitation. I hope to be there sometime soon. It's going to be one of the first things I go to the Academy. Oh, hey, John.
We'll be happy to have you.
John Fong.
Thank you.
Thanks for tuning in. Thank you for the invitation, everyone. Claudia, good to see you.